Hello, everyone, and welcome to this year's 2025 webinar series. We're starting off the webinar series with a ISCN specific webinar mini series of three nice webinars to present the new ISCN updates of 2024. Today's talk will be on the summary of changes, and and today we'd like to welcome Dr. Hastings, who is a state registered clinical scientist and fellow of the Royal College of Pathologists. She has more than 35 years of experience in constitutional, acquired, and molecular cytogenomics, both in research and diagnostic setting in the UK. For the past 20 years, she was the director of an ISO accredited proficiency testing provider for cytogenomics and genomics. And Dr. Hastings now works part-time as a cytogenomics consultant for GenQA, an ISO accredited PT provider operated by Oxford University Hospitals. Dr. Hastings is the chair of the ISCN Standards Committee since 2020 and the editor of the ISCN 2020 and lead editor of the ISCN 2024. Dr. Hastings has authored many publications and book chapters on genetics, NIPT, PGD, ISCN, and PT. In addition, she is an author on many best practice guidelines. And now I will turn it over to Dr. Hastings for her presentation on the ISCN 2024 updates. Thank you.
Good morning, everyone. It's afternoon where I am, so I have to think about this for a minute. Just to say that we're going to just discuss about all the summary of the changes. This applies to constitutional neoplasia, and we're going to probably give mostly examples from constitutional today where they are slightly different from neoplasia. The next talk will be on neoplasia, and the third talk will be on optical genome mapping. So I'm going to just declare potential conflict of interest. And that is because I'm the ISCN Standing Committee Chair, but I'm also a consultant for GenQA, which is a not-for-profit global ISO accredited EQA provider and part of the UK NEQUAS consortium. You refer to EQA as PT testing in the United States. And as the EQA programs support the use of ISCN, I don't think there is a conflict of interest, but some of you may perceive it as such, so I'm just declaring it now. Where there are asterisks, I'm hoping you can see the arrow. This denotes examples from ISCN 2020 or ISCN 2024, and they're shown here with permission from CARGA. They cannot be. If you want to use these examples from the book, you do have to get permission from CARGA to do so. I'm hoping that you've all got the book. If you haven't, you may like to consider. My screen is not moving. That's good news. Oh, it is now. You may like to consider buying it simply because this was a major review and there are a lot of changes in ISCN 2024, and you may ever want to buy the book on online or just the online version. So what I'm going to talk about is why we need nomenclature, a bit of background to the ISCN, some changes that were introduced in ISCN 2024, some educational tools that are available. I won't have covered everything that's available globally. These are the ones I'm aware of. If you're aware of others, could you let me know, please? Because then I'll put them into the talks. The Standing Committee has a copy of these talks. This one's been specially done for the CGC, so it's been split up and extra examples have been given. And if there's time at the end, we will do a quiz. So I hope I don't have to tell you why we need global nomenclature. It's a requirement for accurate reporting of results. It means we can talk about our results without any language barriers, and it allows for accurate interpretation by the clinicians. And it means that patient results can be understood by different healthcare providers, whether it's in the same country or cross countries. Plus, it gives clear communication of results in publications, and it gives consistency for the database entries. I'm getting more and more questions now about when to use ISCN and when to use HGBS. Historically, they came from different areas. the HGBS is more to do with sort of frame shift mutations and nonsense, and it's all about the actual sequencing. But now that we're getting technologies that are overlapping the two disciplines of molecular genetics and cytogenomics, we tend to talk about genomics, then the question then becomes, when do you use ISCN and when do you use HGBS? So ISCN is used to describe variants at a chromosome or gene level, and it includes numerical changes, structural aberrations, pathogenic variants such as CMVs, haplotypes, and repeat expansions. And that will include duplications, deletions, insertions, inversions, translocations. And it describes what you see. So if you've got, for instance, very simple example, trisomy 21, then it will be ×3. HGBS is used to describe DNA, RNA, protein sequence variants. So that's slightly different because ISCN does not cover RNA and protein sequence variants at a gene, nucleotide, and amino acid level. So it's at a very high resolution. And again, you will have insertions, deletions, and duplications, but you'll have frame shift missense and nonsense and tandem repeats and SVs. However, what it does do is it describes a change on one chromosome or one strand of the DNA compared to a reference sequence. So if you had trisomy 21, it would have ×2 because there are 2 additional copies. And that's where there's a fundamental difference between the two. We do work together to make sure we don't create new nomenclature that is in contradiction to the other nomenclature. So just to give you a bit of history about ISCN, the initial 5 publications in 1971 through to 1985 did not include specific neoplasia ISCN. That didn't come in until 1991, would you believe? FISH came in 1995. The first part of array CGH came in 2005, but was very simplistic. Then in 2009, we introduced IDEM and sidelines, which again is very late. Genome build and region-specific assays came in 2013. Sequencing came in 2016, and the term cytogenomic. 2020, we introduced polar bodies, DMAT, DPAT, tricolor probes, complex. And then 2024, we've done a revised structure. We've codified it. So we put all the rules in. They're in each individual chapter, but they're also a generic one in chapter 4. We've introduced genomic mapping nomenclature. There's nomenclature now for moles, for complete and partial moles, for uniparental disomy, for unclear sex, or not unclear sex, that's the wrong thing to say, but when you don't want to disclose the sex, and some nomenclature for methylation-specific assays. So this is the ISCN Standing Committee. 2 of the members have since now stepped down since we published ISCN 2024, and there is a call for new members, but it's from, and I put form there, Asia and Oceania. We are part of the Hugo Standards Committee, and one of the reasons why the price has gone up, it's got nothing to do with the committee, it's obviously to do with the publishers, but when 2020 came out and there was the first online version, it was only available for 1 year. Now the online version is available for 4 years. So this is what it looks like online, and it was released in November last year. Any ISCN errors will be corrected on the online version, but obviously they will be published in the journal for a correction, so you're aware of them for the book, but you can just write them in the book. And then when you go in, there is a forum. Just to highlight this, because some people still don't seem to know of its existence. And this is where you can ask questions. This is a page from it, and you can ask any question you like. You won't get a fast reply. You can look at the previous ones and see what the responses were, or you can, as here, is open a new question. We look at these at each meeting and go through them, and our meetings are monthly, so you can see why we don't always respond quickly. So the ISCN 2024 is still used for karyotypes for FISH, for regional specific assays, RSA, arrays, which is called genome mapping, and for sequencing. So what we've done is we have restructured All the general rules are summarised in Chapter 4. The specific technique rules are in each of the relevant chapters. The ISCN examples are now numbered. This is so you can say to your colleague, it's example so-and-so on, under this section, because before it was you have to count all the examples and it wasn't easy to find when you're trying to compare notes. And the rules are now listed alphabetically. We've also put some additional nomenclature examples in, and the regional specific assay has expanded to include targeted chromosome analysis, targeted microarray analysis, fusion genes, expansion repeats, and methylation assays for imprinting. And there's a new chapter on genomic mapping. And the ideograms are now in the appendix. So don't go looking from the front of the book. This was so you could find them easier. But a lot of people can't find them now. Sorry about that. We thought it was better in the appendix. You could just go to the back of the book and find them. We've used the latest genome build throughout for the band designations and nucleotides, and we try to streamline the nomenclature so it is consistent across the different technologies, and the changes are not marked in the margin because there were so many that Cargo decided it wasn't appropriate for putting the line, grey line in the book, because most of the book would have been grey. So this is just to show you where all the different changes are. Those in red are where all the major changes have been introduced. You will notice there's now 16 chapters, whereas ISCN 2020 had 20 chapters. The meiosis chapter has disappeared, and the variant chapter has now been incorporated into chapter 2. So chapters 4 and chapter 5 have been separated out so that you have got all the structural rearrangements are in the karyotype chapter, and all the rules, generic rules, are in chapter 4. You've got the new chapter on genome mapping, and you've got an expanded region-specific assay one, and the appendix has changed. This is just a quick guideline to show you where all the additional examples are, and you can see it's really across the board, with the exception of perhaps numerical and structural, but there are a lot more examples, and obviously genome mapping with being a new chapter has got all of those examples are new. Unfortunately, even though we tried our very best, in some cases, Cargo introduced some errors which we didn't spot, and in some we actually missed the error that was there. And this is a list of all the errors that have been found. They will be, there's 2 that are not on the website yet, but they will be on the website very shortly. But they have been changed where appropriate. So the big change is there's no spaces, except there has to be a space between the technique and the genome build. And the reason behind this was we wanted to take out the space altogether, but it doesn't work for metaphase fish. And all the examples we had, we just thought people are going to make mistakes. So we've put a space in. So there's a space after RSA, after array, and where I put it in red, that's where the space is new. There is no space now after the copy number and whether it's inherited or it's homozygous. So this is a change where there was a space before Matt, Pat, and de novo, and after the copy number, there isn't any longer. You still have space between 2 symbols or abbreviations, such as pseudodicentric, and you still have a space before and after concept, mosaic, and chi. The big change is we try to explain what the different formats are. So you have a karyotype format and you have a microarray format, and it will depend which one you use, will depend on the technique and the structural information that is given by the assay. So the karyotype format, you will probably be very familiar with the short and detailed systems because you've used them before for banded analysis. It's also used for in situ hybridization. Genome mapping, sequencing, and some RSA can use both karyotype or microarray format, depending what the assay is telling you about the structural information. So as I say, the short and detailed for karyotype format is exactly the same as in ISCN 2020 form. The abbreviated form is for numeration of the fish probes. It was there, it just never was actually specifically specified that this is an abbreviated form of the karyotype format. And I've just given you an example here of the format. Then you've got the microarray format. Again, it's abbreviated short, and we've called it extended because it was called extended before, as in ISCN 2020. Just to note that the abbreviations Peter and Cuta are not used in the microarray format. They are used in sequencing, and that is because HGVS uses them, even though you cannot pick up that it's Peter and Cuta, you're making an assumption because you've got repeat sequences right at the very end. But in a microarray, we've actually specified you can't use it. You also shouldn't be using 1 because, again, you can't pick up the end of the chromosomes. So the abbreviated form you'll be very familiar with. That's just for a normal, and then 3 copies if it was triploidy. Sorry, I said it was normal, but that's for 3 copies of that's a triploidy. The short form where you just put the nucleotides that are lost, and the extended form where you talk about the normal, where the normal last normal nucleotide is. Commas are optional, so you can include them or you can choose not to, but they must not be included in the extended system, except obviously after the copy number. Because otherwise, if you take them out, if you look at it, you won't be able to work out where the copy number ends and where the nucleotide starts. We've also tried to clarify the text from some common misconceptions, which have come from the PT and EQA providers predominantly, and also from the forum when we've realised people don't understand what the rules are. And so for the constitutional, we've put all the ISCN structural chromosome examples into one chapter because they were over 3 different chapters before. We've put for both neoplasia and constitutional, we've put the order of the cell lines when you've got the same cell numbers or the same complexity in neoplasia, and we've said you sequentially apply these rules. We try to make them so they're easy to use and they will work. If you find there is an example of a case that you've got where it doesn't work, will you please let us know, because we've tried lots of examples to make sure it does work. And obviously, human nature will do different things, and we may need to have another rule if you find an example where it doesn't work. We've also changed the undisclosed sex, because when you had 46, X query, where you didn't want to disclose whether it was a male or a female fetus, for instance, we had feedback saying basically, well, query means you don't know if it's really the X, which is true if you look at the rules. So we've changed the query and the X to U, where U replaces XX or XY. The other thing that we have added to the ISCN is metaphase fish in that chapter, or part of that chapter, chapter 7. And that's because when you have a tandem duplication, which is this one here, with the duplication of 17, you can just put ++. However, if you've got a non-tandem duplication, such as in this image where you've got other euchromatin in between and you can see separation of the signals, then the ISCN isn't very clear that that is a non-tandem, or it wasn't very clear. And it came up again through an EQA case. And so now we've put an example in where you have to actually specify the probe separately because it's non-tandem. We've also tried to clarify the fish results. So normal results are given according to the probe sets in all situations. So that does affect CLL probe sets, and it affects the aneuploidy probe sets. So in ISCN 2020, you could do this. However, in ISCN 24, you have to separate the X and 18 from the 13 and the 21, and there's a comma between the 2 probe sets. We've also removed the detail system for all normal results, which includes break apart and fusion probes. So you only use the short system. So the ones that I've crossed out are no longer, you can no longer use. It caused confusion because people thought these were abnormal, and that's why they were taken out. This is ISCN 2020. Now you've got a space and the ×2 goes outside the bracket. So the fish is now consistent with array sequencing and RSA nomenclature. The only time the multiplication is inside the brackets is when there are different signal patterns. So in this particular case, this one is outside because it's the same signal pattern, but this one is inside because you've got a different signal pattern for RB1 and the chromosome 21 contig. The other thing we found inconsistency was we're streamlining, again, across different techniques, about when you have a loss or a gain or inversion in the same breakpoint. When it's karyotype-like, ISCN that you're using, you specify both breakpoints because otherwise it'd be a terminal deletion. However, with array sequence and sequence-like ISCN, so it's the microarray format, you only specify it once, simply because the nucleotides will tell you it is an interstitial deletion, not a terminal deletion. So when you come to the rules for mosaicism, in constitutional, I will talk about a little bit of neoplasia, but most of this will come in the next talk about neoplasia. In constitutional, to indicate a mixed cell line, the largest cell line goes first, but the normal cell line is always listed last, regardless of size. When it comes to clones, it's the least complex acquired clone that's listed first, and the individual neoplastic clones, if they are present with constitutional cell lines, they're listed in the following order. So your abnormal clones with the acquired abnormality go first, then the cell lines with a cell line with a constitutional abnormality goes next, and then the normal cell line, if it's present, goes last. So this is an example, say, for instance, where you've got loss 21, it's in a trisomy 21 constitutional patient, and then it's a mosaic trisomy 21, so you've got a normal cell line there. Then for both mosaicism and clones, as I said, the normal cell line is always listed last. The only exception is nuc-ish because the normal is in the denominator, and so you don't actually specify it, with, again, one little exception, which is for sex chromosome determination. And I've just given you an example of that there. It's in the book. All these examples are in the book. So when you've got, which does happen, I mean, one way to get around it is just do extra cells, but sometimes that's not possible. Where you've got equal number of metaphases in the cell lines, the following sequential order applies. And this is given in section 4.53 for both constitutional cell lines. It's given for related clones and unrelated clones. They're all put under the same sequential rules. So where the abnormalities in different clones or cell lines involve chromosomes of a different number, the chromosome order rule applies. So it is X before Y before any autosome. So 1 before 2, etc. When the same chromosome is involved, so in this particular case, I'm sorry, I better go back to the first thing, rule. XXX goes before 21, and in true chimerism, if you had it, XX would go before XY. When the same chromosome is involved, gains are listed before losses, before structural changes, and we specified that and made a rule about it simply because we had contradictions in 2020 ISCN, where sometimes the gains were put first and sometimes the losses were put first. So we've made a rule to say which way it should go. And the structural abnormalities, if it's the same chromosome, that is involved, then it is the Del's will go before inversions, will go before ISO. If you've got a structural abnormality. When it's the same type of abnormality, so you've got two structural abnormalities, they both involve chromosome 13, then it's Peter to Q rule that applies. So a del 13Q13, Q14 will be listed before a del Q14, Q22. And say they're all in the book, but it's just for me to explain how the rules work. We've also added, and it affects array more than anything else, there are times when you've got a mixed cell population, but you can't precisely quantify it. And obviously, you can use tilde, so that's no change from ISCN 2020. There's no problem using that if you still wish to. If it can't be determined at all, but you know it's more than 2, you can put greater than 2 and then put question mark. And obviously, it would imply also for neoplasia where you want to use AMP, but you can't quantify how much amplification is, so you can put AMP and then put a question mark after it. There is a new acronym now for uniparental disomy. And we have given a table of a tabulated array result because sometimes we get more abnormalities than it's really feasible to put in a long string. Now that may depend on your LIMS or whatever system you have for reporting your results. But if you can put a tabulated result in, we've given you some advice on how to do that. We've also included some chimera examples, some complex array results showing different types of chromoanagenesis, chromothripsis, and chromoanasynthesis. And we've said you can put the normal X or Y in the array ISCN if it gives clarity for the gender. And this is an example of a tabulated complex result. So you have got an example here, which would work for array, genome mapping and sequencing. We are suggesting that you should put the chromosome in, the band, the genome build, the genome coordinates, the abnormality, the type of abnormality, whether it's loss, gain, amplification, region of homozygosity, and the proportion of the sample with the abnormality where applicable. And of course, in the comments, if there is a specific gene that is involved, you can put it in. Obviously, if there's 91 genes, for example, you're not going to list all of them. There may be some that are more clinically significant that you might want to list, but the advice is not for you to look at the whole lot. Excuse me, just seen a problem. That's okay. When there is uniparental disomy, you can now put, we could before put parental origin in, but if you know what it is, it should be designated UMAT or UPAT. And we've also put some chimera and complete mole examples whereby you've got examples for a complete high datitidiform mole where there's monospermic fertilization of an empty ovum. There's one with dispermic fertilization, but no, in both cases, no maternal complement. And when you do have an XX chimera where the haplotypes are different, then the largest cell line is listed first. So we've hopefully given you examples which will cover your clinical scenarios. The big change is that we have added in the RSA some methylation-specific MLPA nomenclature. And so we've got new acronyms which fit in with the HGBS of MET for normal methylation, GOM for gain of methylation, and LOM for loss of methylation. The normal situation, what was happening was we've used the nomenclature that is published to deal with the actual imprinted genes, whether you've got gain of methylation, loss of methylation, or normal methylation, but what wasn't present was the copy number. And so we've now put together the two lots of nomenclature, so you've now put the copy number in, because sometimes you do get gain as well as loss. The other thing that is really changed, and again came as a request from the genomics community, is targeted assays. Other than FISH should use the acronym RSA. And this is to describe normal and abnormal results for specific chromosomes or regions that tested. You can't use the genome build and just put the abnormality if you haven't looked at the other chromosomes, because by implication, you're saying you've looked at the whole genome. And so if it's just targeted for the chromosome or the region, then you use the RSA as your technique, and you don't use ARR. So we've now got this for both targeted chromosome analysis, so that is for when you're perhaps checking in neoplasia or even in constitution when you're looking for mosaicism and you're looking to exclude something and you're looking for specific chromosomes and you're not doing a full chromosome analysis, or for targeted microarray analysis when you're only looking again perhaps in a parent or just because that's all you've been asked to do, looking for a specific chromosome or region in array. So this is when you'd use RSA. And if you've got a numerical or structural abnormality, this is all in chapter 9. Just trying to remember if it is 9 or 10 now. Sorry, you've got a numerical or structural abnormality. For a balanced chromosome complement, where the structural changes, where the homologue is apparently normal, the abbreviated or the short system can be used. It's chapter 10. And the short system includes the breakpoints, but the abbreviated system doesn't. So if you're looking for 11.2 translocation in the parent because there's derivative 22, for instance, in the child, then if the parent's normal in the abbreviated system, you just put RSA 1122 times 2, or for the short system, you put the breakpoints in. And again, in this particular case, it's times 1 because you're talking about that particular abnormality that you've seen it once. If you look at non-rearranged chromosomes, where a homologue is lost, if you're looking at a non-rearranged homologue that's lost, then the short system nomenclature is used with the number of normal homologues described as 0, because otherwise you will assume, if you look at the abbreviated system, if you've got an ISO 21, you're making an assumption that there isn't a normal 21, but there might be a normal 21, and this is a trisomy 21 patient. So you have to put times 0 to make sure it's clear what you're actually seeing. And again, if you've got a 1421 rearrangement, a translocation, not a derivative, you've got a normal 14 and you haven't got any normal 21s, sorry. And that would be, the cell numbers would be given because it's a neoplasia. If you've got an unbalanced chromosome complement, they're always described using the short system, where the normal cell chromosomes are also listed. So if you've got a trisomy 21, it's unbalanced. So you've got to say the 21 is one copy, normal copy 21, you've got an ISO 21. If you've got a 1421, you'd have one copy of the 1421, one copy of the 14 if it was a trisomy 21, again, due to a 1421, you would have 2 normal 21s as well. And for mosaics, you just put a slash line like you would normally do, and you put the cell numbers after it. when you use a targeted array, you do use RSA, not array. So if the first example of a normal here with 22 times 2, this is a targeted array analysis for the whole of chromosome 22. The next one would be a targeted array analysis for the George critical region. And so you'd put the nucleotides that you looked at and say it was normal. And if you got loss of it, you could use the abbreviated short or extended system. But again, you're putting RSA in front, not array. In the sequence ISCN, then we've tried to clarify the terminology. We've given ISCN examples for the different platforms and bioinformatic algorithms. This was a lot of work done by Laura Conlin with some checking of the examples, obviously by the standing committee, and also by the HGVS chair. There are more normal aneuploidy, mosaic and complex examples that have been added. And as this is used and adopted more, there'll be more examples given. I might just add for the whole book that we will never, ever be able to put every single example that you would possibly see in a diagnostic lab. It's impossible because there are so many variations. What we're trying to do in the ISCN is give you the rules so that you can actually describe the abnormality that you are seeing. In the description in sequencing, we have also stated whether you have to use both HGVS and ISCN, so both of those lines have to be included. Whether either could be used, so you can choose whether to use HGVS or you can use ISCN, or where ISCN is more informative. And that's often where there's inheritance patterns where you can say it's derived from something else, because you can't do that in HGVS. Sequencing is now the same as array, that the same breakpoint loss or gain is only listed once, because again, you can tell from the nucleotides. And the normal is just like array or RSA, where you'd put for a female, x, 1 to 22 times 2. If you've got an abnormal aneuploidy, you've given several examples of this. If you had a monosomy X, for instance, you can just put sequence space x times 1. Or because in HGVS, you don't know whether it's the X or the Y is lost, it becomes this little symbol here that looks like an arrow going upwards means or in HGVS. And so you'd have to say either we've got loss of the X or we've got loss of the Y. You can choose which of those two you use. Personally, I'd use the first because it's so much easier and simpler to understand what it is it's saying than I'm very used to ISCN. So I said I'd talk about educational resources. The ISCN book is obviously there. It's also online, so you can go and look up things. You can actually search for things online, which makes life easier. And you've got the forum to ask questions. But if it's a diagnostic case, please don't expect a quick answer. We can't guarantee we can do that. If you're involved in proficiency testing or external quality assessment, there are many national and international EQA providers that include ISCN in their assessments. Some providers, such as GenQA, also have a specific gene ISCN EQA, and there are publications. There is a publication just come out from Cytogenomic Genome Research, which summarizes all the changes in ISCN 2024. So that's online, available now. I can send the link to Celeste and she can send it to you all. There's individual competency modules now through GenQA. It's called Genie, and it's an individual competency test. And there's one release for ISCN, and there's one release for HGVS. I'll mention that in a bit. There are also plenty of webinars you've got and workshops. So you've got the ASHG. We did one last year. We're doing another one this year with some complex cases. There's some on the GenQA YouTube, although we have to update them from 2020, some of them. There's some workshops, again, if you look on the websites for Cancer Genomics Consortium or for GenQA and possibly other places. So just to explain what Genie is. It's an online training and competency tool for individuals. You select the area of genomics where you want evidence of competency. It's modular. So there's many, but there's some for CMVs and SVs. There's some for HGVS and ISCN. And you can do it individually and pay the fee, or you can do it on a laboratory basis as well. And this is currently what they're covering. So there's quite a bit on sampling. There's a bit on BRCA1, 2. There's some on variants. There's a new one just come out on variant and which variants you should be classifying and which ones you shouldn't. But the one I'm going to talk about is ISCN nomenclature. And there's a new trial that's just opened, and there are 3 levels: basic, advanced, and complex. Only the basic is currently available, and it's available in constitutional, neoplasia, and FISH will be released soon. And so what you get if you're doing a karyotyping module is you'll get an image of what the abnormality is, a sort of diagrammatic image, and you'll be told what the abnormality is, and then you're given lots of options to choose from, and you have to choose the correct one. And you get randomly, so somebody in your centre will not get the same 5 or 6 cases, and there are about 50 to 100 cases actually on the system, and it randomly gives you them. You then put your answers in. When you've finished doing it, it's free at the moment if you are interested in signing up. And when you finish, you'll get a certificate of participation virtually immediately afterwards, within 2 minutes. So this was one of the trial results, and you can see that, unfortunately, for some of the variants, the ISCN was not correct. In about 6 out of the 10 of those ISCN scenarios, the correct result was given by 60% of participants. So it's not that high. We would love to see it much higher than that, and they weren't particularly difficult cases. So you do get a certificate of participation. This is one I did just to prove that it's possible to do. And you'll get feedback. You'll get told if it's a correct result, and you'll get told if it's the incorrect result, what the correct result is and why your result is incorrect. So just to thank everybody who've been involved in setting all this up. We have the ISCN Standing Committee, yourselves and the genomics community across the world for all the suggestions. We had more than 250, most of which were accepted, and we changed the ISCN. Cargo, obviously, for doing the ISCN online and making the forum work for us. And some of the data I've given you comes from GenQA, and some of the slides are from Laura, and the rest of them I've done. So I've got questions now. So how do we do this? Right. Okay. So I put the questions on. I can't move.
So I shared the result. If you want to speak about it. Yeah.
So I'll move. Hang on. I can't move my slides now. Can you see my slides still? There we are. So what percentage of laboratories submitted the correct ISCN for GenQA constitutional microarray ISCN in 2024? And the answer was 53%. So the next question is, what do you think the percentage of papers submitted to Research with Genetics in Medicine journal had the correct HGVS nomenclature? Okay, so the vast majority thought it'd be 35%. That's 50% of you. And then the next highest would be 65%, and 15% thought it was 80%. In fact, I can move over. It was 5%. So I shall move to the next question. If you had a prenatal sample of a maternally inherited 1122 translocation, and they did not want to know the sex, which of these is the correct ISCN? Has the poll stopped?
Yes. Yes, sorry.
Oh, right. Okay. So 98% of you got exactly the right answer, and 1% unfortunately chose the wrong answer. A is incorrect because even if you were using ISCN 2020, there should be no space after the bracket. B is ISCN 2020, so please look at ISCN 2024, and well done to everybody else. Next question. You've got a mosaic postnatal result with 3 cell lines. All the cell lines have 15 metaphases. Which is the correct ISCN 2024? It's this little rule, and it's a change from.
Okay, ended poll.
Right, thank you. So 76% got the correct result. Well done. And nobody picked the first one. Well done to everybody on that one. I'm so pleased that we must put the normal cell line first. And with B, it's gains before losses. This is the rule that we've introduced. So it's just to remember now it's 47xxx before 45x. Next question. This is for a non-tandem duplication. Which of these ISCNs is correct?
And I ended the poll.
Okay, so 81% of you got the correct answer. Again, well done. 5% chose A, and the reason that is wrong is because that's for a tandem duplication, and this is a non-tandem duplication. 14% of you said B, but ×2 is only used for normal results in ISH. Obviously, it's also used, ×2 is used obviously in nuke-ish, but this is sometimes where the confusion happens, is people get mixed up between what you can use in nuke-ish, because there is a ×2 for the normal in metaphase-ish, but you can't use it for abnormals. So the next question is, you've got an array that shows paternal homozygosity for the long arm of chromosomes 15 and 21, which is the correct ISCN 2024 for this result.
Ended the poll.
Thank you. Well, nearly 60% of you got the correct result here. Going to move on. There it is. 10% of you chose the first one, which would have been ISCN 2024, but you need to now have a space after the array and you need a U before Pat. 14% of you chose B, but there's no space before homozygosity now, and you again need the U before the Pat. And 16% of you chose C, but the times 2 should be outside the brackets, and you need U before the Pat. So when we come on to targeted array, but well done, that's good. Targeted chromosome analysis for only chromosome 8. It's a mosaic non-disjunction trisomy 8 in 10 out of 30 metaphases, so which is the correct ISCN?
I ended the poll.
Thank you. Again, 67% of you got exactly the correct answer. It's not the first one. Oh, but I'll give you the answers. Hang on. There we are. Because the denominator is used in Nukish, not in RSA. In B, which a few of you thought was correct, you've got the normal cell line should always be listed last. And in C, this is the result for a targeted microarray ISCN. And so that's where the difference is between chromosome and array. And then the last question, now this is slightly different. And now it's which is the incorrect ISCN. There's only one that's incorrect. All the others are correct. So you've got to pick out which is the incorrect result. So this is for an array.
Ended the poll.
Right, last question then. So again, vast majority of you got it right. Well done, everybody. 74%. And this is for some of you will be new. You've done very well. So all the other 3 are quite correct because they're talking about the region of 22q11.2. B is incorrect. Oh, you've all got it wrong. Sorry, that's right. Yeah, B is incorrect. Then you got it right. There's me getting mixed up because it's the incorrect answer. Because it's only talking about the whole of chromosome 22, and you've only looked at 22q11.2. So 74% of you got this answer correct. You correctly identified the incorrect answer. How confusing can you get? So the others, A, C, and D, do actually are the different forms that you can have to write the RSA nomenclature, depending on whether or not you're using the abbreviated, short, or extended nomenclature. So that's the end of all the questions. I'll stop my share now.
Excellent. I would like to
just to be consistent with our thing about Peter and Cuter.
I would like to thank Ross, and I will really quick share my screen. There are more questions, but I will share those with her to think about for future editions, I guess. I would like to also welcome you to come see us at the next webinars. We have part 2, neoplasia specific changes, and part 3, optical genome mapping. And with that, I also want to remind people that we have the annual meeting coming up in wonderful Houston. So early registration ends on May 21st. So please, I hope to see everyone there August 3rd through the 6th in Houston. And finally, if you have an interesting case, we are always looking for people to present at the Wisconsin and CGC co-meeting. And it's a virtual meeting, so it's not doesn't need to require any flights. And it is a wonderful time to present in front of an international audience. So this QR code is here, but also if you have anything, you can email me, or there's places on the website to enter the information.